So you get 2 potassiums coming in, you get 3 sodiums going out each cycle. What do we call this thing?
Sodium potassium pump?
How appropriate. Sodium ion, potassium ion pump. Leave the dash out. You know how to write this now, so you get the answer right. What answer? Okay. This is also called primary active transport. Primary active transport. Active means it takes ATPs. Primary active transport. Did you wonder why there were all those sodiums out there? It's because of this. Where is this pump? Don't be fooled. You got loads of these pumps all over your cell membranes. The skeletal muscle cell, it's got them. Other cells do, especially like skeletal muscle. Okay, so this is active transport, primary active transport. Okay, bear with me a moment. These sodium ions didn't want to go out. In fact, they're mad. They got kicked out. Took energy, but they got kicked out. They're angry. So we come down here on the next page, 75, and we see all these angry sodium ions. It took energy to pump them out. There's the ATP. You pumped them, pumped them, pumped them. And maybe some potassium came in too. But you pumped out the sodium ions, and they are really provoked. They wanted to be in here, they were in here, and you kicked them out. Don't you feel a little bit guilty? No, I'm sure you don't. So here, they're fuming and mad. So the neighbor says, oh, shut up. Why are you making so much noise? And they said, because we wanted to be inside, and we got kicked out. And the neighbor says, listen, settle down. I'll let you come through me. And they say, you will do that? Yes, I'll do that. How much is it going to cost me? They're not stupid. They're checking this out. How much is it going to cost me? And the pink one says, nothing. And they say, oh, that's a deal. Oh, well, that's not a good enough deal. I'll throw in, you can bring a friend with you, a sweet friend. So who are you going to bring with you? Glucose. So who's getting brought in? Glucose. Who's coming in? Sodium ions. And everybody's happy, right? Yeah. What did you just do? You just brought glucose into the cell. And it didn't cost you anything more. That's a deal. But it cost you to begin with, because when you got kicked out, it took ATP to do that. So you did primary active transport, and now you're using that for benefit. And you're bringing glucoses in without any extra energy being needed, but you're getting them in. Sodiums came in, and you can turn around and do it all over again.
And the glucose is going to be used?
It can be used for whatever you want at this point. Just getting it in was the problem, and you just solved
So
it.
it's coming in with the sodium?
With the sodiums, that's right. And this is not primary active transport, this is secondary active transport with the sodiums you had kicked out.
So we're just cycling sodium in and out, in and out?
You can do that. But it cost you originally, so it wasn't all free. Originally, you had to use ATP to get them out, and now they came in naturally. Because there's so many out there, they would come in. And this would allow them to come in. And they were even allowed to bring glucose. So you effectively got glucose in the cell.
Which I'm gonna use to make ATP, which I'm gonna use to take the
Yeah,
sodium out.
you can use it whatever way you want
Right.
to. Isn't that interesting? How that's all connected.
But why did they originally, I understand that they're being pumped out, but why are they being
It's
pumped out?
concentration.
Okay.
Okay. You get,
Is it for homeostasis?
you got somewhat. You got all this sodium ions on the inside. You want to pump them out. You don't want them in there, but there were more on the outside. That's why they came in. You can pump them out. So there is active pumping that goes on just because you needed to do that. Because like you got water coming in your basement. Well, here we don't have much in the way of basements. But if you did, you could put a sump pump down there and pump out the water. And it might keep collecting and you have to keep pumping it out, okay? So some things have to be repeated over and over again, but this is something that happens. You can pump out the sodiums, takes you ATPs, but then you can turn around and use that to bring glucose in. Any question?
The secondary does not require ATP then, right?
No additional ATP, yeah. And it required ATP for the primary active transport, but not for the secondary. And there are places in your body where the cells do this. All right, we're doing pretty well there. I think we hit a lot of the stuff. I'm going to go back to the note sheet to see what we missed. You know about enzymes, right? Say yes, or I'm gonna be irritated.
Yes.
Okay, and you know about ATP?
Yes.
Yes, and you know about the twisted ladder and nitrogenous base pairs, hydrogen bonds, zipping and unzipping, DNA replication, DNA polymerase, nucleotides, DNA and RNA are made of nucleotides. You know about life cycle of a cell, interphase a lot of the time, but then you can do mitotic phase, okay? You have 46 sister chromatid pairs, everybody knows that now, right? Okay, and the sisters are joined by a centromere, and DNA, when you have it, can be wrapped around histones, okay? Chromatin strands, we know about that. Protein synthesis, first part is what? Transcription, and second part is translation. And we know about the Golgi apparatus for cleaning up and repackaging. We know about endoplasmic reticulum, rough and smooth. We've worked on quite a few things here. And RNAs, what kinds of RNAs did we talk about? Messenger RNA, the meat, what else? Ribosomal RNA for the bun, what else? Transfer RNA, the guitars, good, okay. Now there were some strands that we talked about, supportive strands. Who made the mitotic spindle? It was composed of microtubules. You say, when did we talk about intermediate filaments? We didn't until now. Today, we did. Before, intermediate filaments, tell me. Smooth muscles have that netting around them, remember? With dense bodies, intermediate filaments, dense bodies. That's true, okay? And then microfilaments are actually made of actin. Phagocytosis, pinocytosis, receptor-mediated endocytosis. We did that. B snares, T snares, sodium ion, potassium ion pump. We did that. Secondary active transport, we just did that. We brought in glucose that way, okay? We're getting ready to do this stuff probably next time, okay? So we are going to do our, wet lab next time, but we're gonna have a quiz first, okay? And we may go over some new material too for the next quiz, okay? We gotta keep moving because test 3 is gonna be coming soon, okay? And you're doing pretty good. The answers I'm getting show that you're understanding pretty well. So we wanna talk about some stuff in chemistry here that we'll get into, but not so much today. Do you notice how we've talked about things in the past that help us understand this? Adenosine triphosphate, that's not new. You had that already. ADP you had the phospholipid bilayer. You've had a lot of stuff here, but what chapter are we in? This is 3, and some of the stuff was 2. So the beauty is you've had stuff that we can build on, which is why we jumped around and didn't just go chapter 1, chapter 2, chapter 3. Because this stuff wouldn't have meant much to you if we had just gone 1, 2, 3. But we didn't do that. So now it means more to you and it should help you. Okay, we wanted to do that.
So why isn't the book structured that way?
You have to ask them. They choose what they want to do. I didn't write the book, okay? But I chose the way I wanted to do it. So I made it so you would be, I knew what, it's not like I haven't read the book. So I knew it was coming. So I could decide what I want to emphasize, when I want to emphasize it, and then put the story together. So that's what we've done. We've done that. And it gets you ready for some things you didn't know were coming, okay? But that's okay, that's good, that's actually good, okay? You remember your moon landers?
The guitars,
See them, there they are, or the Mars vehicles, those could be Mars, okay? We started talking about that the other day, that's for your test 3, remember, okay? And you had learned some stuff there, you picked up some things, you've learned some chemistry, haven't you? Oxygens, hydrogens, carbons, that sort of thing, dehydration, synthesis, hydrolysis. Remember, when did you first hear of hydrolysis?
About 2 classes ago.
When?
About 2 classes ago.
More than that. Tell me when you first heard hydrolysis. Okay, I'll help you out. ATP hydrolysis, now tell me.
The myosin filament.
Cross-bridge cycle, that's been a little while, hasn't it? Yeah, see, you got that back there, and I knew you were gonna learn hydrolysis at that point and coming across here. And if you're going into AP2, you're gonna meet up with hydrolysis there. You're gonna meet up with apoptosis. Yeah, I'm not telling you what that is, but we will when you do AP2. There's all kinds of goodies coming. It's exciting. If everything was known, everything wasn't exciting, it'd be boring. Are you bored? No? Okay.
I have a question.
Yes.
The, I forgot the call. The
Centromere?
No, not that. The thing that pulls them apart.
Oh, the
The
mitotic
microtubules.
spindle.
Yeah. What is that?
Those are some strands in the cell. See, this is another thing I talk about. Your cell cannot have bones. Bones are way too big to be in a cell, but they have a skeleton. What would you call it? Cytoskeleton. And what makes up this skeleton? It's microtubules, microfilaments, and intermediate filaments. They are the.
Like a scaffolding?
Yeah, they're scaffolding, they're structural supports. I was hoping you'd come back. Okay,
And those are just way too small to like
yeah.
know what they look
Well,
like.
we're going to be talking about them a little bit here. I just looked at it a little bit, I have to look back where it was. See, it's in the cell. It's in the cell, that's chapter 3. See how chapter 3 and 2 go together pretty nicely? They really do. Okay, I'm on page 8. Page 8. Page 8. Top of the page. Look at what we got there. Doesn't that look like you could build stuff? Yeah. See, these are microfilaments made of black olives, right? Actin subunits. So there's something like that in, and look at how good this book did it. Look at it. That's an actual photograph of actin filaments, microfilaments. So I don't know how they got it there, but they got it in this photo. And here, they found intermediate filaments. Remember, they did the lattice work around the smooth muscle cell, and we had it one other place, I'm not remembering right now. It looks like rope. Doesn't that look like rope? And they got a photo of it in these cells. This was good photography. They got a photo of intermediate filaments in there. And here, they actually were able to separate out by just coloring them. They got the microtubules. So they gave, I love this. I mean, you can talk about it, that's one thing, but actual visual, that's good. That's helpful. It makes you feel like it's real. Just like the polyribosome, you feel like it's real.
And the centrioles are made out of the microtubule.
The centrioles, let's see. Where do we have, oh, the centrioles. There, that's a good point. You've got the centrosome, normally have one. But when you're going to do the dance of the chromosomes, you get 2 because you double it, and then you hook these microtubules into these things and make that whole spindle apparatus.
Okay.
And,
So they're like an anchoring.
yeah, it's an anchoring there, and you can take it apart again. The cell can assemble and take apart. So amazing. And nobody's standing there saying what to do. Pretty
You
interesting.
think you can take the centrioles apart?
Yep, well, the centrioles, you're gonna stick microtubules on there to make that spindle. You can build the spindle, you can take it apart.
Okay.
The cell does, listen, nobody's saying do this, do that, do this. It still does it. It's so amazing.
You ever think that they come pre-programmed to do just one function and that's it?
It comes, well, sometimes you use microtubules for different things. So it can break them apart, put them together, and put them together in different ways.
No, I'm saying like, maybe at that level, at that scale, maybe every little structure has its own like pre-programmed instruction.
I
And
don't
they
know
just-
how, I don't know how you pre-program things. I don't know. Maybe the size,
Electrical signals?
like things certainly fit together, that could be part of it. They naturally fall together, maybe, but I don't know.
This book is very simple, so my micro, the illustrations are